Welcome back to HIM tutorials. As nitrogen cycle as a subtopic we are doing under the main heading microorganisms, and the learning objective for today is to learn about the nitrogen fixation. This is the first step of nitrogen cycle and we will learn in detail about the nitrogen fixation. So first of all, what is nitrogen fixation? So converting the atmospheric nitrogen, as atmospheric nitrogen is having 78% of it in the air. So converting this atmospheric nitrogen into the usable form of nitrogen, that is in form of ammonia, nitrite and nitrate. So when we are converting in these nitrogenous compounds, then the nitrogen is fixed, and because we are not using directly the nitrogen, so we have to fix that nitrogen with the help of plants and animals, and then only we can use it for our benefit. So let's go for the nitrogen fixation. In nitrogen fixation, we will learn two types: abiotic nitrogen fixation and biotic nitrogen fixation. Now, abiotic nitrogen fixation means without the help of organisms, and biotic nitrogen fixation means with the help of organisms. As without the help of organisms, again we are going to learn nitrogen fixation in two ways, that is physical or atmospheric nitrogen fixation, and second one is industrial nitrogen fixation. So physical and atmospheric nitrogen fixation and industrial and nitrogen fixation, we will discuss them, but first we will go for the biotic nitrogen fixation with the help of organisms. Under this we will learn about the third type, that is the biological nitrogen fixation. In biological nitrogen fixation, again we are going to learn about two types, that is non-symbiotic nitrogen fixation and symbiotic nitrogen fixation. Again, the symbiotic nitrogen fixation is divided into two parts, and that is with the help of leguminous plants and with the help of non-leguminous plants. So this is the outline structure, that what is there in nitrogen fixation, and I know it is very lengthy process, but we have to go for this in detail like this, then only you will be able to understand that how the nitrogen is fixing into the from the air into the usable form, that is in the ammonia, nitrite and nitrate. So nitrogen fixation has two types: abiotic nitrogen fixation and biotic nitrogen fixation. Abiotic is without the organisms, which is divided again into physical or atmospheric nitrogen fixation and industrial nitrogen fixation, and biotic nitrogen fixation is with the help of organisms, which is again having biological nitrogen fixation, and it is divided into two types: non-symbiotic organisms and symbiotic organisms. And in case of symbiotic organisms, again we are going to learn about leguminous plants and the non-leguminous plants. So let's check first about what is there in physical or atmospheric nitrogen fixation. As there is no organism involved in it, but still during the lightning, what happened? There is increase in temperature and pressure in the atmosphere, and we know that there are two gases as nitrogen and oxygen are present. Nitrogen is present in 78% and oxygen is present in 21%. So what is happening here? The nitrogen and oxygen, they combine to form nitrogen oxide. Again this combine with oxygen and they will form the nitrite. Again this nitrite will combine with the oxygen and it forms the nitrate, and this nitrate will again combines with the water. When the nitrate is formed in the air under the high temperature and pressure, and soon there is lightning, thunderstorm takes place, and the water is also there as a rain. So with the help of rainwater, this nitrate will combine and it will form the nitric acid. Now this nitric acid come on the earth as a form of acid rain, and when it comes on the earth in the form of acid rain, then this nitric acid combines with various type of minerals, for example calcium, iron, and they will form nitrates of that mineral, just like calcium nitrate. So all in all, what is forming at the last? Nitrate is forming at the last, as the atmospheric nitrogen is converted into the usable form, that is the nitrate form. Now next is the industrial nitrogen fixation. So for the industrial nitrogen fixation, we will learn it with the help of two processes. First is Haber's process, and second one is the Birkeland-Ides process. Now what is there in Haber's process? In Haber's process, the atmospheric or you can say the hydrogen and the nitrogen they combine with each other at very high temperature and pressure, because it should be in artificial conditions, because it is one of the part of the industrial nitrogen fixation. So under the artificial conditions, high temperature and pressure is there, and hydrogen and nitrogen, they are combining with each other and they will form the ammonia. So again the nitrogen is converted into the usable form, that is the ammonia form. In Birkeland-Ides process, there are three processes. First is arc furnace, second one is the oxidizing chamber, and the third one is the absorption chamber. So in arc furnace, again nitrogen and oxygen, they combine and form nitrogen oxide. Again this nitrogen oxide combines with the more oxygen and it will form the nitrogen dioxide. Now this nitrogen dioxide combines with water, because it's absorption chamber, so it will absorb water and it will convert into nitric or nitrous acid. So in industrial nitrogen fixation, two parts are there, that is Haber's process and Birkeland-Ides process. In Haber's process, what is going on? Hydrogen and nitrogen At high temperature and pressure, they will convert into ammonia. And in Birkeland-Ides process, in arc furnace, nitrogen and oxygen combine to form nitrogen oxide. Then oxidizing chamber, nitrogen oxide will combine with the oxygen to form nitrogen dioxide. Now this under the absorption chamber, this nitrogen dioxide, it will combine with water to form the nitric acid or the nitrous acid. And again, the nitric acid is what? That is the usable form of the ammonia, usable form of the nitrogen, which is fixed to form many kinds of proteins, amino acids and nucleic acids in our body. So this is the discussion about the abiotic factor. But when we go for the biological nitrogen fixation, so it's a little bit such type of process in which the organisms are involved in the nitrogen fixation. So in biological nitrogen fixation, it is of two types, non-symbiotic and the symbiotic. In case of non-symbiotic organisms, mainly certain bacteria and algae help in it. So the first is some aerobic bacteria. Aerobic means that will work under the presence of oxygen. So aerobic bacteria second is anaerobic, that is in the absence of oxygen. So aerobic bacteria is Azotobacter and anaerobic is Clostridium and Rhodopsirillum is also there in books. And third one is the blue-green algae or cyanobacteria is also it's also called as cyanobacteria. You will also got the name of nostoc in it and Enamina. So these are the organisms which will help in fixing the atmospheric nitrogen, and they are non-symbiotic. Now what does this symbiotic word mean? Symbiotic word means the organisms are in mutual, two organisms, they will live with each other in mutual relationship. They will mutually benefit each other without harming each other. So this is the non-symbiotic one, and in case of symbiotic, we will discuss about the leguminous and the non-leguminous plants. So in case of symbiotic, again it is divided into two main parts. Leguminous plants and non-leguminous plants. So leguminous plants or non-leguminous plants, before discussing about them, first we have to go for that what is leguminous plants or what is legume. So legumes are somewhat that which have nodules in their roots. As simply the roots are like this structure is having of the roots, and in the roots these leguminous plants have this type of circle, nodules, which is called as, they have nodules in their roots. Now in these nodules there is presence of some bacteria. So the bacteria is living in the nodule and it will fixing the atmospheric nitrogen for the plant, and plant is giving protection to that bacteria. So in case of leguminous plants, we will go for all kind of pulses. So we will take example of pea, gram. So let's have example of pea. In pea there is presence of rhizobium bacteria, and leguminous plants we are discussing. So we will discuss again two types. This is the root legume. As a root legume means in pea the legume, the nodule that is present in the root. So P is having rhizobium in the root nodule, and what rhizobium is doing? Rhizobium is making nitrate for the P, and as a result P will give protection to the rhizobium. So the second part is the stem legume. This is very rare. Very rare plants are there which are having these nodule-like structures in their stem. This is somewhat like this. This is the stem, and they have this kind of nodules are present in the stem. And in this we will take example of Sesbania. And in Sesbania there is presence of bacteria called as azorhizobium. Now what azorhizobium is going on, what function he is doing? He is just fixing the atmospheric nitrogen into the nitrate, that is a usable form, and the Sesbania is giving protection to the azorhizobium. Some non-leguminous plants are also there which also help in fixing the atmospheric nitrogen into the usable form of the nitrogen. So here we can take the example of Elnus, and Elnus has the bacteria called as Frankia, and this Frankia is fixing ammonia in form of nitrate, and Elnus will provide protection to the Frankia. So this is all about the process of nitrogen fixation. I know it's a somewhat lengthy process, but to understand all the things we have to go like this, then only we will be able to understand that how atmospheric nitrogen is fixed by the different kind of organisms under different categories. So nitrogen fixation was divided into two parts as abiotic factors and the biotic factors. In abiotic factors we will go without the organisms. Then there are two main types, industrial nitrogen fixation and atmospheric nitrogen fixation. In industrial nitrogen fixation we have learned about the Haber's process and Birkeland-Ides process. In case of biological nitrogen fixation, it is with the help of organisms, and it will again divided into two parts, that is non-symbiotic plants organisms are helping and symbiotic organisms are helping. Non-symbiotic has aerobic bacteria, example azotobacter, anaerobic bacteria which work in the absence of oxygen like Clostridium, Rhodospirallum, and some blue-green algae, cyanobacteria, nostoc, amebina, these are the examples in it. Then in case of symbiotic symbiotic organisms, leguminous plants and non-leguminous plants are helping. So in leguminous we are going again divide into two parts, that is the root nodule, root legume and the stem legume. So in case of root legume, the P is there which is having rhizobium, and rhizobium is fixing a nitrate for the P, and P is giving protection. Again in stem legume, the Sesbania is there. It is very rare but still it exists. So Sesbania is fixing atmospheric nitrogen in form of nitrate for the azorhizobium, and azorhizobium is having protection from the Sesbania. In case of non-leguminous plants, Alnus is there, and Alnus is again having bacteria Frankia in the nodule, and Frankia will fix the nitrogen in form of nitrate, that is usable form, and Alnus will give protection to the Frankia. So this is all about the nitrogen fixation, that how nitrogen fixes. There is little bit more in it, we will discuss it in next session. So I hope you understood all very well, and our objectives also complete, that we have learned about the nitrogen fixation deeply. So up to our next session, keep watching, keep learning. Thank you so much.